Welcome everyone to the last Grand Challenges Initiative lecture of spring semester. I'm really glad that everyone could join us. And I'm really glad to hear from Ronke Olabisi, who is just down the road at UC Irvine. And unfortunately, she's joining us by Zoom. We'd love to have her in person. We'll invite her back after you see how awesome all of her research is. You can pester me and we'll find another time. She is an engineer in the Samueli School of Engineering there. I asked her before we started the seminar what her favorite undergraduate class was when she was at MIT, where she studied mechanical engineering, and she gave an answer that I almost don't want to tell you, because there's absolutely no way we have a class that matches this. So don't hold this against me. But when her professors were explaining parabolic flight to her in an aeronautics class, because all of the professors had their own planes, all of the students just went up in the planes and flew around. This is like the ultimate in experiential learning. And now I will go look on Amazon to see if I can buy a plane so that I can fly you all around, or maybe like eBay or something. So without further ado, I'm going to cede the floor to Dr. Olabisi, and hold on to your questions. You can drop them into the chat, and at the end, we'll save some time. Those of you that have attended GCI lectures before know that we have a rule that within the bounds of civility, no question too weird or odd. I'm looking forward to hearing also what's on your mind. Without further ado, Dr. Olabisi, thank you for joining us.
Thank you for having me. So I'm going to give you a brief overview of the main research that's going on in my lab. And so, and then I'm going to focus on one project. And so here I do some wound healing, but I do wound healing using these microencapsulated cells. And you can kind of see that one set of wounds has healed very quickly by day 14, whereas different types of cells don't heal it as fast. And I put them in cells so that in these microspheres, so that they don't have to, you don't have to have immunosuppressant drugs. And when they're in them, like if you imagine grapes inside of Jell-O and you imagine ants as the immune system trying to get to the grapes, the immune system can't get in. So it's like a force field that protects these cells so that they can keep doing what they're doing. And here, the green are cells that are still alive, and these are cells that have died. They're stained red. And yeah, so those work well. They're dead now, because though there are more that are dead now, because I think this is 3 weeks out. I also do a project on retina repair. And so in the developed world, the #1 cause of blindness is age-related macular degeneration. And these are retinal epithelial cells. And so your retina is essentially like an epithelium is a skin layer. And so it's a kind of epithelium, and it's very delicate, and it lives on this thing called the Bruch's membrane, which is kind of like, it's the basement membrane for the retina. And it's also kind of like a bed. And the retina is going along doing fine, but then this bed, it's kind of like it gets bed bugs in it. And so when that happens, the retina that is sitting on it will start to degrade in those spots. And so somebody who has age-related macular degeneration, the macula is the area in your eye where all the light gets focused. So in that spot, it'll be like they have like a dark spot right wherever they look that follows them around. So they can't see directly where they're looking, but they still have peripheral vision. And so as you can imagine, you can't watch TV or read or do any of the things that you want to do. And so we're investigating ways to help repair that Bruch's membrane. In addition, I've worked on bone repair, and I'm going to be talking about the bone repair. This is one of the ways that we worked on it, and this was bone that was formed from cells that were not encapsulated, cells that were programmed to make bone, and from cells that were encapsulated. And what happens is that the immune system wants to take it away. And so that's why this is small. And when you have them encapsulated, the immune system cannot. And so I'm going to talk to you about the ways that I borrowed from the ancient Maya to form bone. And because you guys are all young people, I'm going to give you a preview or a teaser trailer first that I did for TikTok. And I don't know if the sound will work, so I'm going to hold my microphone over there and then play it.
What do bone, seashells, and rock candy have to do with each other? They're all made of crystals. In rock candy, it's sugar crystals. In bone, it's hydroxyapatite, and in seashell, it's calcium carbonate. And the thing about crystals, like this rock candy I made here, is that they like to grow on something. We call that a nucleation site. For the rock candy, it's string. For the bone, it's a protein called collagen. But we wanted to see if we could tell bones to grow where we wanted, so we used tiny seashell proteins to make tiny crystals and to put those crystals into patterns, and it worked. The bone grew exactly where we wanted it to, in those lines there. Not as delicious as these crystals, but still pretty cool.
Okay, so you'll have to forgive me for the corniness. It was for younger students in middle and high school, but still, it kind of gives a teaser of what I'm going to talk about. And so in the United States alone, there's over 500,000 bone grafts that are performed annually, over 2 million worldwide. And bone grafts are required when there's an insult to the bone that exceeds its capacity to repair itself. And this can occur in fracture when the gap is too large, for the bone cells can't cross that gap is too large. And so you're going to need a bone graft to help repair it. So it occurs when you have a fracture. It occurs when you have disease like osteomyelitis, which is an infection in the bone, and it causes a gap that's again too large to repair. When you need to fuse the spine, so like you would fuse the spine to repair the scoliosis. Here the surgeons would first straighten the spine, and then they would fuse it to prevent the curve from reoccurring. If you have a spinal fraction, so here, like to repair this fracture, you want to fuse the spine, and you need a bone graft to do that because the spine won't fuse on its own, or to repair a herniated disc. So here, this disc is herniated. And so if you are not familiar with what the vertebral discs are like, so it's the shock absorber of your spine. And you can imagine that it's like a dollop of snot wrapped with a fruit roll up all the way around. And so that fruit roll up, if it gets a tear in it, the snot is going to leak out. And you can't sew a fruit roll up, so you can't repair this. So the more of that snot is going to keep pushing and pushing and pushing and causing random pain in everything that is lower than this spot. And so the way to repair that, and this is a simulation, it never looks as good, is to scoop out that disc because you can't repair it, and then try to cause these two bones to fuse together. And so this is great when it works, but the procedure it fails 50 to 70% of the time, which is a high rate of failure. And so the grafts, when we talk about the grafts used for all of these procedures, they can be from a variety of sources. And there's the autograft, which is an autologous graft, which means it's taken from yourself. So usually they take these from different parts of your body. The allogeneic graft, which means it's taken from a human donor, so another person. The xenogeneic graft or xenograft, that's a donor from another species. And so that's what these are trying to show. This is coral. Or alloplastic grafts. These are non-metallic, polymeric, or ceramic grafts. And so the autograft right now is the current clinical standard of care. It doesn't cause an immune response because it's from you. It'll form bone wherever you put it. The problems are that it requires an additional surgery to harvest, right? And so if I'm taking bone from that hip, I've just created what's called a critical size defect. You're not going to repair that. You need a bone graft to repair that. So now you're missing this piece of bone. Or they can take it from the scapula, which is the shoulder blade. But you know, you can only take so much to repair your bone elsewhere. And patients have reported pain at these harvest sites up to 2 years post-operatively. So if you have a big giant defect that's larger than what they can harvest, then the procedure is not going to work for you. And so because of this, there's this pressing need for alternatives to the autograft, right? And so any alternative, it has to be as good. It has to be osteoconductive, and osteo means bone, and conductive means that it can conduct bone, so bone can grow on it. It has to be osteoinductive, meaning that it can induce stem cells to differentiate into bone cells. It has to be osteogenic, meaning that once it's differentiated, the bone cells will actually build bone once they're there. And it's got to be bioresorbable, in that the body can degrade it. And so to understand the strategies for bone repair, you have to understand how bone repairs itself. And so I kind of made this little cartoon. You have to forgive me because it's handmade if it's not good enough. So let's say you have a break. So first, what happens is bone morphogenetic protein is released from the bone, and it causes the chemotaxis and differentiation of mesenchymal stem cells, which are also known as adult stem cells. And these will differentiate from stem cells into chondrocytes and pre-osteoblasts, right? And chondro means cartilage and cyte is a cell. And so chondrocytes are cartilage cells, and they come in and they lay down this cartilaginous callus. I'm gonna make my animation go, and that stabilizes the bone. And then the pre-osteoblasts are gonna differentiate into osteoblasts, and these osteoblasts, you know, osteo meaning bone and blast meaning build, they'll build bone using that callus as a scaffold. And once they're finished, they release receptor activator of nuclear factor kappa B, which causes single nuclei monocytes to fuse, and they become multinucleated osteoclasts. And again, osteo is bone, but clast means to cleave or to cut. And these osteoclasts release hydrochloric acid to degrade the mineral part of the bone, and cathepsin K, which is a protease, to degrade the organic part of the bone. And they reshape the bone into this proper anatomical shape. And so the thing that's really pretty cool about bone morphogenetic protein type 2, so I love science and engineering history, it was discovered that in 1965, this researcher named Marshall Urist, he discovered that if you took demineralized bone and you injected it into muscle, it would cause bone formation. And then in 1971, he figured out that it wasn't the bone itself, it was the proteins within the bone that were responsible. And then it wasn't until the late 80s that the first bone morphogenetic proteins were isolated. BMP1, BMP2, and BMP3 were the first. And then since then, over 20 have been found. BMP2 is FDA approved in certain procedures, but it has a lot of off-label use, meaning surgeons, it's not approved for it, but surgeons will be like, oh, that'll work here. I'll use it off the label. And it's BMP2 is FDA approved. No matter where you inject it, it makes bone. You inject it in your thigh, it's gonna make bone. You inject it in your cheek, it's gonna make bone. So the problem is that it's delivered in a solution. So if it leaks, you're gonna make bone where it leaks, right? And so it's, even though it's only FDA approved, because in certain places, because it makes the bone so well, like I said, there's a lot of off-label uses for it, but the off-label uses have started a lot of these complications. And so this is what it looks like when it's injected intramuscularly. So the mesenchymal stem cells will do the same thing. They'll differentiate into pre-osteoblasts and then osteoblasts. Well, that's the pre-osteoblast, I'm ahead of myself. And then the osteoblast, and then they're gonna lay down bone. But because it diffuses rapidly and it's not attached to anything like bone fragments, there's this limited bone formation response. And so because the bone formation response is very low when you inject it, very high doses are required to get enough bone to repair anything, right? And so if a clinician is using BMP2 through injection, they use 1,000 times the amount of BMP2 that is found in your entire body just to get the bone to form and to fuse. And so as you can imagine, this causes complications, right? there have been so many. It's a growth factor, and high doses of a growth factor can cause cancer in people who are prone to developing cancer. But even in people not prone to developing cancer, bone growing where it shouldn't is a problem, right? And so here, so MRIs, they show soft tissue very well because they show the hydrogen ion, and the hydrogen is abundant in water. So you see things that have a lot of water. So CT shows things that are radio dense. So they show bone very well. So this black is the bone that is white in the CT, right? And so we can see that here, this is the spinal cord. Like this is a vertebral body right here. And this is just a zoom in. A and B is far away so you can see where it is, but C and D are zoom in. And this is the vertebral body. And this right here is the spinal cord. And there's fluid that is developed because of the BMP, and it's pressing on the spinal cord. Now, you can't see the spinal cord as well in here because, again, soft tissue, better seen through MRI, but you know that this is kind of the same place. Also, you can't see the metal that they've put in to screw because, again, soft stuff, better on MRI. But this is ectopic bone, and ectopic means it's outside of where it's supposed to be. And the spinal cord is right where that ectopic bone is. And so people are getting neurological impairments, they're getting difficulty breathing because, you know, it's messing with the nerves that are responsible for all of this. And so what's the problem? Well, the problem is that unless you use an autologous graft, then all of the grafts available require BMP2 because they've been, well, specifically the allogeneic grafts have been processed so that they don't cause an immune response, and the processing kills everything in there that causes bone to form, right? Okay, so now you understand how that grows. Now, let's understand a little bit more about what happens when the osteoblasts and osteoclasts are doing their normal job. So no matter how old you are, you guys are late teens, early 20s, doesn't matter. Your skeleton is at most 10 years old. And so every single one of your body parts has a different age that it turns itself over. And so one of the fastest things that turns over is your hair cells and your stomach. And so like that will turn over like every 24 hours, you'll get new ones of those. And this is also, as an aside, why you lose hair and weight when you have chemotherapy, because chemotherapy doesn't just target cancer, it targets rapidly dividing cells. Now, the rate at your bone turns over is 10 years because your osteoblasts and osteoclasts work together to continually turn it over so that the wear and tear you put on it doesn't accumulate. Because otherwise, if you imagine taking the tab of a soda can and you bend it back and forth and back and forth and back and forth, eventually it breaks. That would happen to your bones if they didn't turn over. And so you form these little micro cracks just by walking around and being your happy self. And your body is like, okay, she's got some micro cracks, let's fix that. And what that looks like is these osteoclasts are like, I am going to eat up a bone that has those micro cracks. And the osteoblasts are like, I am going to replace it with brand new bone. And don't mind if I do, don't mind if you do. And so when you have osteoporosis or when you're an astronaut in space, what happens is that the osteoclasts are like, I'm still going to do my job. And the osteoblasts are like, okay, catch you later. And what happens is that you have 1 to 2.7% bone loss per month in astronauts, which is 10 times the rate of postmenopausal women who lose it at a rate of 1 to 2% per year. And so it's comparable to disuse from paralysis or bed rest. And so it's not that people with osteoporosis have weaker bone, it's that they have less bone, right? And so you can see this is a normal bone matrix, and this is osteoporotic bone, which has a lot less, right? And so there are some countermeasures for bone loss. And so people use bisphosphonates to kill the osteoclasts. There are non-medicinal uses like exercise, nutrition, red wine, resveratrol in red wine has been shown to help. Having low animal protein to potassium ratios and lifestyle changes. And as I said, medicinal approaches and hormone therapy like estrogens in postmenopausal women and other drugs that are used to kind of build bone and help prevent the loss. Now, the problem, so bisphosphonates were like, dude, we'll just kill the osteoclasts. They're the problem, right? And so in osteoporosis, when you just have regular osteoporosis, the fractures happen usually from a fall. Like an old lady usually falls down and she breaks her hip, right? And so this is the hip and this is the top and this is where it happens. And so it's because, you know, their bones are really thin now. And so they gave them bisphosphonates in the hope of helping. But the problem is that they weren't seeing these same kinds of falls. They weren't seeing people breaking their bones from a fall. People would be like, just, you know, I'm great. I don't have osteoporosis. I'm going to exercise. I'm going to walk. I'm going to do all this stuff. Hey, my leg is sore. And then it breaks. And so the breaks would typically happen where there's high cycle loading and the accumulated wear just led to a sudden break when the person is just standing or walking. And so this kind of caused, like, this is a typical bisphosphonate break. And so the only way to fix this is what they discontinue the therapy. And it's controversial because it happens probably in about 2 to 5% of people, but doctors have this do no harm thing. And so while on the one hand, they were helping people who had osteoporosis break, they were helping a potential break, but causing almost a definite break in some people. So some doctors, they won't use it because they don't want to cause harm, even though they might be preventing harm. It's a thing. But in any case, if BMP2 is unsuitable, what's the alternative? Because you can't target BMP2 to these little spots. So, and you can't inject massive doses near the bone just to get them to be fixed. So there's other BMPs that show promise in terms of using them in autograft procedures, but that lack of side effects, it really could be because they're not as widely used, so you don't get as many side effects. And so, but one of the things, you know, your body has 20 BMPs for a reason, over 20 BMPs for a reason. It doesn't like using things as monodimers, right? And so that's what we're delivering when we're delivering BMP2. When we combine it as heterodimers, like BMP2 plus BMP7 together, they're more potent and they require less doses. There's also other proteins that are osteogenic, and there are other molecules that will inhibit the bone resorption without killing off all your osteoclasts. And there's also strategies to control how quickly the BMP2 is delivered. So if you absorb it to a scaffold, then it's not going to diffuse away, so you don't have to give 1,000 times what's in your body. You can modify scaffolds to have a higher binding affinity for BMP2, or you can deliver it with other growth factors like vascular endothelial growth factor to improve its efficacy. And there's also strategies to covalently bind the BMP2 to substrates. And the benefit is that the BMP2, when it's bound, it won't diffuse away from the scaffold like a sponge, but it might decrease the bioactivity of the scaffold. And so it's a trade-off, right? And some of the groups that control these spatial temporal control strategies, try to control where it's delivered, they formed these inkjet printed scaffolds. They patterned BMP on one side. I believe it was like, you can see on this side, BMP2 on one side and inhibitors on the other side, and then they'll put it into a bone defect, and this is the bone defect. And so bone where there was BMP2, it formed in the defect, but not where the inhibitors were. And this is a strategy, and it is a valid strategy, but we wanted to see if we could get similar spatial control without using an inhibitor. Like if we could just say, grow bone here, would it grow bone there, right? And so a lot like the, you know, giving it a piece of string, like the rock candy. So polyethylene glycol, it forms, you can use it to form these hydrogels, and we call it PEG because we like acronyms. And they're widely used in tissue engineering. They're cross-linked polymer networks with physical properties that can be tuned to the desired stiffness, the desired permeability, or a variety of other properties. And so they're bioinert, they're highly hydrophilic, They resist protein absorption, and they have low cell adhesion, and they're so bioinert that they provide immunoprotection to encapsulated donor xenogenetic cells. And what we did, we conjugated the protein. So we can conjugate our proteins to a heterobifunctional PEG. And what that means is that we have a PEG molecule that has a functional group on either side, and those functional groups are different. So on one side, it has an acrylate group so that it can bind into PEG diacrylate, which is our hydrogel. And on the other end, it has something called a succinimidal valerate group, and that allows us to form a peptide bond with our protein and incorporate that protein into our hydrogel. So once we have this substrate, we needed osteogenic proteins. And so we knew, okay, BMP2 is one osteogenic, remember, bone growth protein that we can use, but we want another protein that doesn't have the complications of BMP2. So we looked at seashells, and seashells are made entirely up of calcium carbonate. The outside is this crystal polymorph of calcium carbonate called calcite, and the inside lustrous, shiny, iridescent part is called mother of pearl or nacre. And so scientists and engineers and researchers will call it nacre. It's a polymorph of calcium carbonate called aragonite, and there are similarities in the way that mollusks mineralize calcium carbonate shells and vertebrates mineralize hydroxyapatite shells. So this is the patterns in nacre, and these are patterns of bone laid down by stem cells in vitro. Okay, so I don't know why this is a double one. Okay, so here, this is two different kinds of nacre. On this side, this is what we call bivalve, and so bivalve, it means that the shell has two sides to it, and gastropod, which is kind of like a snail. And sheet nacre forms flat like this, and it's found in these bivalves. Gastropod nacre, it's kind of called a stack of coins because it looks like a stack of coins. Now, the thing that is important to know about sheep nacre is it's described as having a brick and mortar motif. And when you look at it, it kind of looks like a brick and mortar, right? And column nacre is described as having a stack of coins motif, and column nacre is not very strong, like you could probably break it with your hands, but sheet nacre is 3,000 times stronger than synthetic or geologic aragonite because of this structure, which is on the nano scale, right? And this structure is controlled by protein. And it all goes back, the reason that people look into all of this, it all goes back to the Maya, because the Maya were some serious rock stars. So this is 2018 picture of some tooth bling. This is 700 BC picture of some tooth bling. So because the Maya did it so well, these jewels stayed in for thousands of years, right? But before the Maya did this, before people understood that the Maya did this in living people, they thought it was part of funerary rites, you know, to make your corpse look super pretty. And so that's what they thought when this skull was found. And so again, I love the history of this. So in the 1930s, these archaeologists found this fragment of a Mayan skull, and they found that it had these three teeth, this one, and this one, and they're made of nacre. And they figured, oh, you know, the person must have lost their teeth in life, and as part of the funerary rites, the Maya placed the teeth in some gaps so the person could be buried whole, right? And in 1972, because this was in the Harvard museum, this dentist called Dr. Bobbio asked if he could x-ray the teeth, because he was like, that looks too good to be postmortem to me. And he was right. So the teeth showed osseous integration, meaning that the bone had grown into the nacre teeth, and so they were fixed, rigid in there. And like, if you know somebody who's had a tooth implant today, you probably know somebody who's had a failed tooth implant. So this outlasted the person for thousands of years. The Maya performed tooth implants better than what we have today, right? And so when people discovered this, this sparked a ton of research, particularly with this French group led by Dr. Eveline Lopez. And they took a nacre chip and a bone chip and laid them down 1 millimeter apart on a layer of mammalian osteoblasts, you know, which are bone cells. And on the side nearest the bone tissue started to form. On the side nearest the nacre started to form. And so this shouldn't have been able to happen, you know, evolve in two separate groups, right? So even more research was conducted. And it has been called an implantation material of unparalleled virtues. It's very strong, it's very osteogenic, it's biodegradable, it doesn't cause an immune response, and it seems to have all the benefits of autografts and BMP2 with none of the drawbacks. It was shown that the water-soluble matrix of nacre was responsible for all of these benefits, and the same proteins were thought to precisely control nacre's architecture. And it's successfully been used with no immune response in humans, dogs, sheep. They used some to repair in the 90s some maxillofacial defects, the defects in the jaw. They mixed the blood and nacre powder and slapped that in the jaw in France, and they were able to repair some of the jaws. So we were like, okay, sold, nacre, right? And so we looked at several bone proteins that had been described as capable of nucleating bone formation, so nucleating the crystals that are the precursors to bone. And we looked for proteins that were identified in nacre as also being capable of nucleating nacre proteins, so aragonite little crystals. So just like the string nucleates sugar crystals, we wanted these. Now, we wanted the proteins, but we also wanted to bind these proteins to our hydrogel, so we bound these proteins, right? And so with BMP2, there was conflicting data on whether it can actually nucleate hydroxyapatite, the mineral that bone is comprised of, but the dentin matrix protein and bone ciala protein are known apatite nucleators, so that's why we used. And we figured that if pegylation removed the protein's ability to nucleate minerals from solution, it would also remove its ability to form bone. So then we got to work. Oops, that's not what I want to show you. We got to work. We took some shells, we ground them up, we got the powdered nacre, we mixed it in water, the water soluble proteins come out, we collect that, centrifuge, discard that pellet because we just want the proteins. We freeze dry, which is also called lyophilize, and then we combined it with some PEG, succinyl valerate, which puts those proteins onto our PEG molecule. And then when we looked, we wanted to, this is our ninhydrin assay, and what ninhydrin will do is bind to free amines on your proteins. And so we got super high efficiency with RGDS because it only has one free amine, and you want all of the one free amine to be hit, but you want about like 10 to 20% on your proteins because you don't want every available site because you might reduce its functionality. So this, while it looks like this isn't good, this is exactly what we wanted. And so what we then did, we took each protein, we absorbed it to glass size directly, so these are protein on glass, or covalently bound it to the class via a PEG linker, and then we conducted what is called a nucleation assay. And so you put it in a bath with your phosphate on one side and your calcium on the other side, and you run a small current that will prevent non-specific nucleation. And what we did when we looked at it under the scanning electron microscope, the images showed surface features that were consistent with calcium phosphate mineralization. And so all of the unmodified proteins, these are not with no PEG, they all showed some mineralization. So you kind of can't see it very well here, but if you were here, you would see it. The controls showed no mineralization. So it's not just having any protein that does it. So this is blank. This is just the acrylate group. This is bovine serum albumin, which is a blood protein, and it's not known to cause nucleation. So it's the fact that these are, bone proteins that are causing the nucleation. But even cooler is that these are nacre proteins, and they're causing the nucleation. Right? And so we quantitated that, and what we found is that non-pegylated versus pegylated, we wanted there to be kind of like a match so that it's not reducing it. So everybody was fine. All the proteins were happy with being pegylated except dentin matrix protein, which was like, this sucks. Right. And so we didn't get any crystals formed from dentin matrix protein once we pegylated it. Now, one of the interesting things that we found is that there were these submicron features that kind of look like snow on the pegylated water-soluble matrix side. So when it's unmodified, there were more of these features for the bone proteins, but for the nacre proteins, when they were pegylated, there were more of these features, right? So this is the nacre protein, unpegylated and pegylated. There's more of these. This is BMP2, unpegylated and pegylated. There's less of these. So we're like, huh, that's interesting. I wonder if it's important. It is, and you'll find out later. Okay, so next we covalently bound our proteins to hydrogel sheets and seeded the sheets with mesenchymal stem cells or osteoblasts. And so first you have to polymerize your hydrogel, which is photopolymerizable, like you shine a light and it polymerizes. Next, you put your proteins on top, and then you put the slide to make the protein spread everywhere, and then you shine it with light again, and then you seed your little osteoblasts, and then you say, okay, osteoblasts, do your thing, make some bone, right? And so first of two things we did. So first we studied with mesenchymal stem cells, because remember we said we want this thing to be osteoinductive, and mesenchymal stem cells can be triggered to differentiate towards bone. And if that happens, then when you stain them, they will, with this thing called alkaline phosphatase, if they stain a purpley color, it means that they are differentiating towards bone. And as you can see, the BMP2 and the nacre were able, all of them really, were able to cause differentiation towards bone, and the control you can barely see because it did not. Right, okay, and so now this is alizarin red, and these are actual bony nodules, so mineralized nodules. And so when you see these little red spots, it's red stained calcium nodules. And the unmodified BMP2 and the nacre showed, they showed mineralization relative to the other ones. But the only one that showed more after being pegylated was the nacre one. So we're like, dude, what's going on? Okay, so we wanted to see whether or not we could repeat this in 3D, right? Because all of this stuff was 2D. And so we took our hydrogel precursor solution, we took a cell suspension, we vortexed in mineral oil to create this water in oil, vortex emulsion, right? And when you get this emulsion, then you shine white light on it and it polymerizes it and it entraps your cells. And we've got these two phases. We've got the aqueous PEG phase and the mineral oil phase, and these have photoinitiators in both, and that's why we get such pretty little microspheres. And then again, after 21 days, we did alizarin red staining of the encapsulated osteoblasts, because into these osteoblasts, we also put our conjugated or pegylated or unpegylated proteins, right? And so the mineralization appeared to increase with immobilization for all proteins. So this suggests that when they don't diffuse away, it's better. So BMP2 is just like, it doesn't really need to be there that long. These were both, right about the same. Now, bone cielo protein, now dentin matrix protein, that benefit from being conjugated or pegylated, and this is a nacre peptide, and this is a nacre water soluble matrix. Now, one thing that you notice is that in this one, in the nacre ones, it's significant, like the BMP2 and this one are significantly higher, and that's why they have the asterisk than the others. Control didn't really form any bone, it was down there. Okay, now we wanted to induce spatial patterning on a very small scale, so we used a nylon mesh and a photomask, and we repeated these nucleation studies. And what we found, you can kind of see if I go back and forth, right? So the only one that forms patterns was the PEG water soluble matrix. So the BMP2 doesn't look like that pattern. The PEG, DMP1, they just were like, yeah, wherever you want to go, go. And so we quantitated what was within the pattern and what was outside of the pattern, and it was significant. And so then we said, okay, we did this with just solution. So we just formed these crystals from solution. What can we do if we add osteoblasts, which form bone, to the mix? So we formed these patterns on our hydrogels by using a photomask, right? And then we seeded with the osteoprogenitors, either the mesenchymal stem cells or the osteoblasts, right? Okay, so that's what our photomask looked like. This is what our hydrogel looked like. So we took a picture of the hydrogel to prove you know, we're not cheating. There's nothing on that hydrogel. It just looks blank. But we combined the proteins with fluorescein so we could visualize where they were on the hydrogel. And this shows where they were covalently bound. And after we seeded the hydrogels with osteoblasts, these black areas that follow that curve, that is staining calcium deposits. So then we quantitated, well, where in our pattern are these black stains appearing? And they're mostly appearing within the pattern. So then we repeated these studies with mesenchymal stem cells and pegylated water-soluble matrix from Nacre and BMP2. Like you can kind of see when I put that over that they're kind of following that. So they're differentiating within the pattern. And we quantitated that. So for water-soluble matrix and BMP2, it showed that they both have higher differentiation within the pattern than outside of it. And as well, I'm trying to hurry so that I don't go over. I'm almost done. But as well, we did cellular mineralization, and we found that when we quantified that, you can kind of see, like these are alizarin red stains, you can kind of see these little curves again. But again, it's really only in the water-soluble matrix. So we've got these patterns here, and we've got these patterns here. And so this is showing red stain, and this is showing mineral nodules. Okay, so in summary, what we found, we were very excited. All of our unmodified proteins induced acellular and cellular mineralization. When you pegylate BMP1, it can't do anything anymore. The pegylated Nacre water-soluble matrix is the only one that caused the submicron mineralization. And we think that submicron mineralization are teeny tiny nucleation sites that are important for osteoblasts to grab onto and say, I'm going to build bone here. All of the proteins, whether or not they were pegylated, induced the cellular mineralization. We saw that inside the microspheres. But only the pegylated Nacre water-soluble matrix caused the bone formation within the patterns. And it's the only one that did it within the patterns when there were cells present or absent. And we think that because it was in the patterns when it was absent, or when cells came, cells followed that pattern to form the bone-like substance. And so, yeah, the only PEG water-soluble matrix could control the mineralization and could control the water-soluble matrix and the BMP2 could control the stem cell differentiation. That shouldn't say stem cell mineralization, it should say stem cell differentiation. Okay, so what's the big deal? So the big deal is that this is really the first time that anybody demonstrated microscale pattern biomineralization, so we're super excited about it. We published a paper, and we got a lot of excitement. It compares a wide variety of growth factors on a reproducible PEG-CA scaffold, which is important because PEG has no inhibitors, no other biosignals. So we know that everything that is happening because of our protein. N16N is a Nacre peptide that had been shown to cause aragonite formation, but never shown to be able to cause bone formation. And so this is the first time that we did. And it kind of combines the two fields, like osteogenesis and biomineralization. And so it's like, this is how you guys can marry. And so we're very excited by that. And we're excited that this might lead to a potential osteoporosis treatment that doesn't rely on killing the osteoclast. And so a lot of groups have used antibodies to label this thing called tartrate-resistant acid phosphatase, or TRAP. And it's an enzyme found where osteoclasts eat out the bone. And so if we could link that same antibody with some of our water-soluble matrix from Nacre, or even BMP2, we might be able to tell the bone to go over there where it's needed. And with that, I would like to end, and thanking all of the people in my lab. They are the ones who do all the work.
Thank you very, very much. I'm going to invite folks to add their questions to the chat. but I'm going to start perhaps with a question that's not focused on the details of the work, but rather on the idea that you have a lot of degrees in mechanical and aeronautical engineering. I
Yeah.
counted 4, but you're a long way in some ways from doing what I think most of the students on the screen would think of when they think of mechanical engineering. For example, a lot about biology. Could you just tell us a little bit about how you got from, parabolic flight all the way over here to thinking about mineralization?
Yeah. Okay, so I have always wanted to be an astronaut. And so I think a lot of people, when you always want to be an astronaut and you're very young, you think that you have to go into aerospace or aerospace. And so when I started, I was an aerospace engineer. And, I just didn't love it. I wanted to be on the ship. I didn't necessarily want to be designing the ship. So then I switched to mechanical engineering. And I had this design class and I loved it. And in the design class, like we had to make these devices that would enable paraplegic and quadriplegic people to independently ride horses. And it was really fun. And I was like, oh, I like design. That's what I like. And there's limited design that you can do with spaceships. They have to fit certain tolerances. Otherwise they just delaminate and explode, right? So, then when I went to grad school, so I graduated with a degree in mechanical engineering. Then when I went to grad school, I was like, I miss aerospace. So I went back to aerospace. And then I had a professor who I was taking a composites class in an aerospace department. And he said, I want you to design a material for a non-aerospace application. And I had no idea what to do, so I was just surfing the web. And I came across this hip implant that had broken through somebody's leg. And the more I read about it, the more they described that it was because of a mismatch in material, that the material of the hip implant didn't match the bone. And the more I read about that, the more I discovered that bone, like I had been programming all of these optimization software for my materials to say like, okay, it needs to be bigger here to optimize under that load. But bone does that naturally. Like bone is so cool. And the more I researched bone, the more I fell in love with it. It decides whether or not it needs to be there. It will resorb itself because it thinks it doesn't. So you're in space and it's like, we've got all this extra mass that you don't need. And so it starts. So I absolutely fell in love with bone, and that's why the break happened. And so I was doing more and more and more and more biomedical engineering. And then when I switched to biomedical engineering because like I loved bone in a way that I didn't love anything. And so I was like, okay, I want to love what I do or do what I love, right? And so like if you can't do what you want to do, then you got to figure out a way to love it. And so, but I found the thing that I loved, right? And so I moved to biomedical engineering. And when I graduated, there was a National Space Biomedical Research Institute, and that married everything that I love. So that married space, that married bone. The loss of bone is a huge problem for astronauts. Biomedical engineers are really sorely needed in space because like you do need the people to get to build the ships that get you into space, but then you also need the people to, if you're going to be looking at, human space flight, the people that keep the people from like dying of radiation or loss of bone or all of these kinds of things. And so, I'm still heavily involved with space because of that. I love it.
Probably most people on the screen know someone who has had, for example, a hip replacement or some surgery that's used to compensate for wear and tear over time. And maybe you could talk a little bit about why that happens, given that you have turnover in bone formation on, that's happening at a slow rate, but continuously.
So usually, like, there's two problems. If you are somebody who's going to have joint problems, you're probably not somebody who's going to have osteoporosis. So things that are good for your joints generally tend to be bad for your bones, and vice versa. And your joints, it's when you get a hip replacement, when you get a knee replacement, it's usually because the cartilage is gone. And cartilage is this funny thing that it can't be repaired. Like the vertebral body and the vertebral disc in the back, it's cartilage, and you can't repair it. You can't sew. I mean, anybody who's seen a chicken bone and the thing on the end, it doesn't have it gets its circulation not through blood pumping through it, but through, like, imagine it's got synovial fluid, and the synovial fluid is like, it's the juice that it lives on. And like, imagine you have a sponge and you step on it, and that sponge gets squished, and then when you release, the fluid flows into that. That's how it gets the majority of its nutrients and waste exchange. And you find that things that have a reduced blood supply, like a cartilage doesn't really have any, do not heal very well. And like, that's why they replace the knees, not because the bone is bad, it's because the cartilage is gone, and now you've got these two bones grinding on each other and wearing each other down like friction. And that's the sad reality, and that we're trying really hard to replace cartilage, but it's really hard.
Okay, there's a question from the audience about translation. So tell us a little bit about how you go from experiments in the lab to say clinical trials, or and what that process looks like.
So anytime you find some discovery, it's probably it depends on the discovery. It depends if it's a device or if it's a drug. But if it's a device that doesn't have to go in people, you could probably get that very quickly. If it's a device that has prior art, like it's the same as something else, probably 5 years, and you can get an exception. If there's a global pandemic and you have a vaccine, very fast. But for most discoveries, like something like this, which would probably, it's either classified as a drug or as a device, right? And so mine would be a device that's delivering kind of like this therapeutic, so it would probably be classified as a drug. You have to have a series of animal studies to show that it's not going to kill people. It doesn't I love the animals and I feel bad for them, but you cannot put a drug in people with 0 animal studies. So first they expect you to do in vitro, which are like the cells that you try them on, in vitro studies, so that you can minimize the use of animals, because if that killed all my cells, you're not going to put it in an animal. So first they want small animal studies, because that's where you do the largest number. And then for the most part, they want larger animal studies. And then after the larger animal studies and you haven't killed anybody, then they will allow it, like there's phase 1, phase 2, phase 3 clinical trials, and they'll allow it in phase 1, which is like healthy participants. And phase 3 is the participants that it's intended for generally. So generally, it's the ones that have the disease or disorder that you are trying to treat. But the first group is demonstrating that if I am healthy, this will do nothing bad to me. Oh, I suddenly can't hear you.
No, that's my fault. No matter how much time I spend on Zoom, the mute button is always omnipresent. I have time. I think if someone in the audience has one more question, you all are being unusually shy, which makes me suspicious. So if no one's got a question, then I'll just take the liberty to ask another question here. Maybe you could tell us a little bit about what drives. I'm fascinated by the fact that there's so much variation in the appearance of these building blocks in our body. I think, as a biologist, I often think about cells, right? And there's lots of different variations in cells, but then to see these other mineral formations that look so they look like bricks, or they look like discs. Could you tell us a little bit about what drives those shapes?
Well, so like I actually modeled that in MATLAB once, and the proteins control the thickness. And so like that's why we're really interested in the proteins. But also and this is partly why nacre doesn't cause an immune response. So bone's got cells all within the bone, and that's why you get an immune response when you have it from a cadaver donor if you don't process it. But nacre is it's acellular, right? And so all the cells are outside of nacre, and it has this like liquid crystal that comes up through the center and through these little pores. And then they just spread out until they meet someone else, and then they form all these little plates. And so when you cut it and polish it, then you see that brick formation. But if you were to break it, it's more like jagged plates on top of each other in different directions.
Cool. All right, last question's coming from the audience. It seems apropos. What do we do to prevent this wear and tear, or more broadly, bone disorders in the future, other than perhaps not spending many, many years in space?
So up until you're 35, you can build your bone bank, and then you can't really anymore. So if you're having, you want good nutrition to build your bone bank, to have as much bone as possible. And I mean, there are genetic factors involved as well. So you're less likely to get osteoporosis if you are male. You're more likely to get it if you're white female or Asian female. If you're African American, you're less likely to get osteoporosis, but more likely to get joint disease or arthritis or things like that. And so your risk varies depending on your ethnicity, also on your weight. Very thin people are more likely to get osteoporosis. Very heavy people are more likely to get knee problems. And so what are the athletes' chances in prevention? So athletes often jack up their joints. It depends on what kind of sport you are doing, right? And so impact sports, especially when you are younger, so keep in mind, jogging and running is considered an impact sport. So that is very healthy for your bone, but it's lousy for your joints, right? So it's this trade-off. And so if I knew all of this stuff when I was younger, what I would do is figure out what my risk is and then go based on that. So I'm very low risk at developing osteoporosis. But at developing joint disease. So instead of doing a marathon like I did, and then I have a knee issue after having done that, I would have swam instead. And I was on the swim team. So somewhere in my brain, I had that right, but I wanted to do a marathon. And so, nacre is in shells, all shells. So the shell that I use is from the South Pacific, but if you see a snail, that snail has nacre. if you see like oyster shells, it's just going to be different types of nacre. So like if you eat mollusks, if you go on the beach and you pick up some shells, and some nacre is prettier than others. I think I have some nacre here. I'm going to show you. I like this one. It's very pretty.
I think perhaps some people know nacre as mother of pearl, or am I making that up?
Yeah, nacre is mother of pearl. It's just like when you're talking about it in a research setting, everybody refers to it as nacre.
As nacre, yeah, yeah.
Yeah.
Thank you so much. This was fun and different and really compelling and interesting to see the different scales and types of experiments that you're doing, which are quite different from a lot of things that are going on here on campus. And we're looking forward to hosting you in person in the future. I know everyone else is joining me in a virtual round of applause. And I wish everyone all the best for the rest of the day and for this marathon meets sprint finish for the end of the semester.